Hi, my name is Rudy Gozlan. I'm a professor in conservation ecology. Tonight I'm going to talk about fish, not fish on its own, but fish in the perspective of conservation. I'm going to talk about the world preservation in the context of how we can sustain developments and maintaining a sustainable biodiversity. And I'm going to talk about you, because nothing is more important than you. Thank you everyone for coming tonight. It's a pleasure to see so many familiar faces. I'm going to talk tonight about world preservation, but I'm also going to talk about you, because what else more important than you? And I'm going to talk about, first, about fish. This is what I'm studying, and that's what I am. So, but I'm not going to talk about fish in itself. I'm going to talk about fish in the perspective of conservation. Although fish in itself is very interesting, and I could talk about fish all the time, but Matthew said we have only 1 hour. Did you know that in the last 10 years we discovered that fish had personalities, that fish had a very sophisticated way of communicating through complicated molecules, and they discovered that fish could feel pain. So you see, it's a kind of vast field where I could speak for a long time. But tonight I'm going to talk about fish in the context of conservation. This is a map of world rivers throughout, which have been published in last September in Nature. In red, you got the hotspots of threats to biodiversity, freshwater biodiversity. And as you can see, the state of play is not really great. There is a lot of pressure on our freshwater systems throughout the world. Well, these threats are generally habitat destruction, pollution, flow regulations. You have issues with overfishing, even in freshwater systems. And you have also the issue of species introduction. Among all these threats, tonight I'm going to focus on one particular threat, which is the issues of invasions and how that would affect biodiversity and how that can be mitigated. And I'm going to try to share with you what I've learned in the last few years on that topic. So why do we look at this topic from a theoretical point of view? I mean, it's always interesting to see that populations which have been introduced in very low numbers, so you've got a very low numbers of fish introduced, suddenly start to establish very large populations. So what we're interested in understanding is how can we establish large populations from a very low numbers of fish. We can also, interestingly, looking at what are the traits of these species, what make them so successful at being an invader. Or also, from a genetic point of view, how can a small population with a very low genetic diversity can establish very large populations going through what we call the bottleneck effect, which means that a very low genetic diversity establishing large populations. So very strong theoretical backgrounds to it. But on a more practical aspect, what we're interested in is looking at what are the entry routes, where they're coming from, how they're coming in our waters. Have they got any ecological impact? Should we worry? Do we have any effect on our native biodiversity? We're looking at the economic or impact, or we want to evaluate the risk to be able to forecast what we should do with it. Should we manage them? And how do we mitigate that kind of issues? So just briefly, I'll show you, these are the different continents throughout the world. And here you got in this axis the number of fish which have been introduced in the different continents. And these are divided into the different ecosystem services that are bringing these fish in the different part of the world. So as you can see, all across the world, the main drivers of these introductions are the aquaculture systems in black here. But also the ornamental systems, so people who have aquariums and introduce fish for aquarium and then release them in the wild. And then you have other sources like angling and sport, especially in Western countries. What is the situation in the UK? Well, in the situation in the UK, when you look at you go in the wild and look at in our rivers, the number of non-native fish that you find in our rivers, so the number of fish which have been introduced in our rivers and establishing in the wild, are directly proportional with the numbers of fish you're importing in the region of England. So In each region, the more fish you import, the more likely you will have them escaping and finding them in the wild. And in fact, it's even more strongly correlated to the diversity of the contingents. The more species diversity you introduce in the region of England, the more likely you will have one escaping and one establishing in the wild. So we understand now where the source of introductions come from. But what we're looking at is, okay, they're coming in, they get established in our rivers, and what we're interested in is looking at if they have an impact in our systems. Do they have an impact in our system? Because at the end of the day, if they don't have an impact, maybe that's not such a big deal. So in terms of ecological impact, we're looking at issues like predation. So do they predate on our native species? Do they compete with them for food, for example, or for spawning when they reproduce? Do they hybridize with our species? So do we lose some integrity in terms of gene pool within our native species? Or do they bring disease with them and transmit disease to our species, and so resulting in the decline of biodiversity? But really, what we're looking at is not single issues in isolations. What we want to look at is change. A fish got introduced in a system, and then you got an ecological change which leads to a discontinuous change in your ecosystem functions. So you go from one state suddenly to a next state, and the system is significantly functioning in a different way. It becomes eutrophic or functioning significantly differently. But when you look at the literature, in scientific literature available, well, most of the studies are very initial effects. People reporting species have been introduced there, but have very long follow-up, are not that much presented. So they cover a very short period, and they don't really understand, or they don't study necessarily very well the processes. What are the processes? Are there any factors which make them impacting our system? And what are the implications for the structure of our ecosystem and the function of this ecosystem? And we don't want to look at just a single trophic. Say, for example, I introduce predators in a system. Yes, of course, it's a predator species, so it's going to eat other fish species. That's what predators are. And so we're basically having a single trophic level. We're looking at just at this trophic level. And if we look at the prey population, they will eventually decline. But what we're interested in is looking at indicator of change on the system, on the functioning of our system, not just interspecific competitions. We want to focus on the major changes that we can find. So there are so many introductions around the world. To prioritize our efforts, we really need to concentrate on these major changes. Change in the food web structures, in the structures of how the species interrelate to each other. And we want to find them as an indicator of change in the way the system and the ecosystem function. So to illustrate that, I'm going to show you a few case studies which are picked up around the world, just to illustrate that aspect. The first case studies is from the African lakes. A little fish, Limnothrisa myodons, is like a sardina type family, is a planktivorous fish. So its feeding habit is on the planktons, little organisms who live in the body of the water. And they have been introduced in the Congo, in Tanzania, and in Mozambique. And the idea of introduction was to develop fisheries. A lot of these countries rely on the source of proteins, and fish are a good way of getting there. So it has been introduced in these lakes and has supported very successful fisheries. So it's a very strong ecosystem service in these places. And of course, because it feeds on the mesoplankton, it feeds on these planktons, then it will deplete the planktons, but it is not detrimental to the functioning of the lake. And you could wonder why, because at the end it's changing the plankton structure and the community of planktons. But in fact, when people have started to look at the way the food web was structured, it is controlled by bottom-up processes. So things coming from the nutrients from the lake, not from the top. So if you're adding another predators at the top, controlling this plankton population, it doesn't have any fundamental effect on the structure and the functioning of that lake, which is controlled from bottom-up processes. The other example, it's an example in Lake Victoria. Maybe a lot of you may be aware of this, which has been much more publicized. It was also introduced as an experiment, but also to develop fisheries, to help people having source of proteins. And they introduced a fish called the Nile Perch. It is a very large fish. It's very voracious, as you can see. And it was portrayed as a fish which have virtually extinguished almost the stock of native fish in this lake. And in fact, the group which has been the most affected after the introduction of that fish is the aprochromine species, which is a cichlid species. And it's very important in these lakes because these are, they call the Darwin pond. They have a very special evolutionary history which make the diversity of these species particular to these lakes. And so losing these species make us globally lose a lot of biodiversity in this type of families. But when you look at the story and the follow-up of the introduction a little bit more carefully, you can see that because it has been introduced and it was very successful at producing large population, then the number of people coming and fishing in the lakes have increased. They want everyone to have a share of this kind of new gold mine. And so the number of fishermen have increased, the pressure of fishing have increased, the number of nets have increased, the size of the nets have reduced to maximize the number of catch. And also that has an effect on the aprochromine species. So an additional change to the fact that the Nile Perch was introduced in the lake. Then to prepare the fish, a lot of trees around the lakes have been cut down, so quite a strong deforestation around the lakes to be able to prepare and smoke the fish. And as such, in these places, during the rainy seasons, a lot of nutrients are washed out into the lakes with algal bloom in the middle of the lakes, or invasion of another invasive species, which is the water hyacinth, who, as you can see, could in part cover the lakes. So a big input of nutrients as a result from the lakes. So you could see the cascading effects. And in fact, if you look at it a bit further, in this area, because it brought a lot of wealth and economic prosperity, then people have come in from a different part of the countries, and AIDS have also been increased and positively correlated with the Nile Perch introductions. Gun trafficking have also been linked with the Nile Perch introductions, and even prostitution. So as you can see, the pictures is not as simple as having a large predators introducing in the lake. It has a cascading effect on a wide range of topics and part of the society, socio and economics, which also will link to a decline of biodiversity. And if you look at in other parts of the world, in Sierra Leone, for example, you can replace fish by diamonds. In Honduras, you could replace them by bananas, and you could go in Libya and Nigeria and Angola and have the same issues with crude oil. So it's not directly an issue with the fish. It's not an ecological perspective from the fish only. So when you ask me what is the real contribution of the Nile Perch to the decline of the aprochromine species, well, it's difficult to say. It's very difficult after to tease out which factors are the most dominant in the decline of this diversity. The third example I'm going to show you, and these are a little bit more detailed because it's a topics I've been working on for the last few years. It's an introduction of fish from Asia, mainly from China. And in fact, in the 1961, in ex-former USSR, there was a lot of propaganda in the way things were run. And all the fishing clubs and fisheries were run by the Communist Party at the time. And so there was a desire to promote development of fisheries, but in the same time to be in line with the party's thoughts. So they introduced and promoted the farming of that fish, which is Chinese fish. It was grass carp. It also comes from certain part of Russia. So it was considered by the party as a good communist fish. It grew large. It was very big. It could feed the people. And it was eating grass, so it was kind of an ecological type of fish. And they promoted that against eating other marine species and started to develop a lot of the grass carp aquaculture. And I found back some of the initial pictures of the aquacultures in Romania in 1961, who developed these first farms of grass carp just by the side of the Danube River. And so they introduced carp from China, quantities, and as you see, I mean, the way of farming is fairly simple, fairly crude. But at the same time as they introduced that species to grow, this communist fish, then they also introduced that Chinese fish, which is called maisuyu. It is a very tiny fish, so they didn't want it for feeding the people. They wanted it as, this is an accidental introduction along with the grass carp. It is it's a very small fish, it's a cyprinid fish, so a type of family of fish, very small. It has a very early maturity, so after a year it can reproduce. It has a batch spawning, so it spawns more than once in the season, so it can have a maximum survival of their eggs. And the males guarding the nest, so there's a maximum survival of the offspring when they come out. They have very short longevity. So it's in fact, it's like a weed. It has a very early reproduction, highly reproductive, very short lifespan, and then it disappears. And it has a status at the moment of the most invasive species in Europe, and one of the most invasive in the world. Just quickly, because I can't help it, I'll just show you fish. This is a female of that species, and as you can see, it's got a very nice yellow coloration, and this is a male with a very dark black coloration. These are the ones guarding the nest. So if you see the dispersal, as I said, they were introduced first with a contingency of grass carp from China, and you could see them here in Romania, Belarus, and there was some in Hungary and Lithuania. So they introduced them along there. And then I'm going to show you how they disperse from these points. So here you got the eastwards introductions, and then slowly they get colonized and transferred from aquacultures to aquacultures and start spreading all along up to the west and start filling up all the main catchments around the west. So as you can see at the end, this is a pictures which was 2009. Most of the big catchments of big rivers all around Europe has been colonized by the fish. And if you're here, you're looking at the profile. These are all the European countries where the fish has been introduced. You could see the profile of colonizations, here being the numbers of fish records, and here are the decades, so the timelines. You can see that they have a very fast, like a weed, very fast exponential explosion of the population, and then it stabilized after. It started to saturate all the waters where it could get. Except when you look at here in the UK, we are still in a phase where we're growing, so we can still do something. It hasn't filled up all the UK yet, so we are not there. And if you look at Spain, it's in the same situation. It has just started to colonize. It hasn't completely filled up the countries. And if you look at here, this is dispersal distances, but it's a log of the distance in kilometers. Here we've got very long distances, so although it's a tiny fish, it managed at the beginnings to be introduced, as you've seen on the map, from very, very far away, because it was moving along with contingency of fish, which were moved by humans. And then slowly, the distance of colonization gets less and less, because when it was introduced after in most of the European countries, then it started to disperse and refill all the gaps in between the different rivers. So distance were getting smaller. Rapidly, I just want to show you the traits because that will have an importance after. When you look at, this is the age of the fish, that's for the female and that's for the male, and here we're looking at the latitude. If you look at traits like the length of the fish, you could see that between different populations, every line is a different population, there is quite a variability in length and growth. And in the same time, you can look at age at maturity when they get mature along the latitudinal lines. You've got also variations with latitude. So it's a fish which is what we call plastic. It has capacity to adapt to different places and to different environments and have this kind of variability along his dispersal. So we looked at the genetic structure of the populations in China. Every color corresponds to what we call an haplotype, a genetic type for the fish. And some populations got a mix of different genetic type. But you could see here is Taiwan. There is a type which is only found in Taiwan. And in Japan, there is one, although found in the mainland China, the other one is only found in Japan. So we managed to have an understanding of the structure of the populations. Our fish that we have in Europe have only been introduced from China. So only from this kind of group of fish and this group of fish. The others haven't been yet introduced in Europe. And when we look at the genetic diversity, I don't want to go too much into the detail of that, but here we got two measures of genetic diversity, the haplotype and the nucleotide. And so if you have a very diverse gene pool, then your population will be here. If you have a very low gene pool, then your population will be here. And in white, you have the introduced populations here. And in black, you have the native populations, which are there, apart from this and that one. And so, in fact, when you look at what I said at the beginning, how can they establish large population from a very poor gene pool? Here, we don't have that. We have a very high gene pool. It is completely counterintuitive, because for a long time, we thought that's what happened. Species arrive in very low numbers, very low gene pool. But now I know more and more studies, and our studies have shown that, is that the species get mixed up. There's more than one introductions. And they get mixed up from different catchments from the native population. So, in fact, we're increasing our gene pools. Here we have a gene pool which is much higher than most of the native population. So we are mixing the different population and increasing that. So, in fact, they overcome very quickly these genetic issues by having more than one introductions or admixing. So that's what I'm saying here. With our genetic studies, we found that they were introduced, although in the very small locations, either as a single location or a small geographical location. When they have colonized, they didn't have so much of a problem because the genetic diversity of these fish were artificially made up by mixing population from the different catchments within China or here in Europe, and have allowed the fish to expand from there. So what are the bioclimatic constraints of that fish? I'm just very quickly show you a theoretical understanding of that before we go into the illustration of it. Here we got trait scales. So for example, we can take a trait like reproductive success. We're measuring how successful it is at reproducing. And we have two species, S1 and S2, and we got a thermal gradient, so from very cold to very warm. And this is the environment's optimum. Yeah, we are in this population. So all the populations have evolved and evolved to their optimums, to develop their optimums in line with the environments. These species, as you can see, have a very wide spread. Although the optimum is the same for the two species, it goes from very cold to very warm, while this one has a much narrower range of adaptations. So if you move that species, these two species, outside in a new environment, like you do when you introduce species outside the native range, then this is the new environment optimum in the new environment where they go. And then you could see these species here, because it's outside the zone of comfort in terms of plasticity, they're non-adaptive. They don't adapt and they got extinct. While these species, because they have a very large plasticity in terms of adaptation to temperatures in this case, then they will very quickly evolve and re-establish new population adapted exactly to the new optimum. And that's what we're looking for in our population. Have they done that? Have they, after being introduced, been able to quickly adapt and readjust to the new environments? Because you have to see, when you see the map, they've been introduced to northern Poland, very cold in the winters, very warm in the summer, to Algeria, where it's virtually warm all the time. So very, very contrasting environments, and they still have adapted to it. So in order to do that, we went to China last June. We had a little expedition there to be able to collect and manage to get materials from all these populations, from the northern part of China to the southern parts. In fact, we went from Beijing, we went right down to the Vietnamese border, and then right up down to Mongolians, to the Russian borders, then we went to the North Korean and gone back again. So we tried to cover the maximum climatic contrasting. In the south, you have tropical type climates, which is virtually 10 degrees and above all year round. And then in the north, you've got very, very cold continental type things with -40 in the winter and +30 in the summer. So very contrasting climatic area. And in our fish family, that the ones we're studying, we know that they start their growth when the temperature is above 12 degrees. So 12 degrees C is a very important temperature limit, below which it doesn't grow, above it, which it starts to grow. And what we did, as I showed you in the graph before, we looked at the reproductive success of our population that we find in China. And we put them along with the number of months they had above 12 degrees. So each population which have 5 months above 12 degrees, then we looked at their reproductive success. And as you can see, there is a spread around, say, for example, 7 and 8 months of above 12 degrees, where you got the maximum reproductive success. Below that, you got very low, and above that, you have a very low as well. And what we did is we projected throughout the world the number of months above 12 degrees. So in yellow, you have the 0 months above 12 degrees, so it's cold all year round. And in red, you have the 12 months above 12 degrees, so it's tropical. You have between, in the winter, I think it doesn't go below the 10, 15, something like that. In the summer, it doesn't go very much higher than 22, but it's always above 12 degrees. So what we did after that, we looked at the positions of our populations we find in Europe and the ones we find in the native range. And in fact, you can look at it and you can find that there is kind of constraint, climatic constraints of the distribution of the fish. The fish are basically locked in between 9 months and the 5 months above 12 degrees. There is a climatic constraint, although it has a potential to go further, it will be constrained by this climatic envelope. And if you look at in Europe, although it has been introduced in Algeria, even this part of Algeria, it's fitting within the 6 or 7 months above 12 degrees. It's not the warmest part of Algeria. And if I go back to our genetic map from our population, and as I say, this is very important to understand the genetic structures of the native populations, because as remember I said, we have only introduced fish from China, not from Japan, but not from Taiwan. And if you look at Taiwan, Taiwan is actually in the tropical zone. It is 12 months all year round above 12 degrees. So, and Thomas Gudgeon's flourishing here, no problems, but it is a special genetic type. So you could imagine that if we have Thomas Gudgeons coming from China, imported from China, so adapted to that kind of climate, the potential for colonization is much, much greater than we have at the moment. So it's important to understand not just the type of species, but also the genetic makeups to be able to understand the full potential in terms of invasion. So you will say, yeah, it has been introduced everywhere, it's very invasive, but does it have an ecological impact? And that's really what the politician wants to know, that's what the public want to know, and that's, as an ecologist, we also are interested to understand. So, as I said before, remember this little fish that you introduced and doesn't have any effect on the food web structures because it was controlled by bottom-up processes. In this case, we looked at what is the effect of introducing that species in the food web. For that, we look at something called stable isotope, C13 and nitrogen 15. We look at the ratio of them to be able to identify which level within the water columns the fish are feeding and what are the source of energy. So the carbon indicates if the energy comes from terrestrial input or from aquatic input, and the nitrogen gives you an indication of their trophic levels. And here we have our native group of species. We got roach, bream, and carp. This is before introduction of that species. And then you introduce the topmost gudgeon, and you can see a shift in the source of energy, but also in the trophic positions of our 3 native species. So you got a shift in our communities, a significant shift in the trophic structures after the introduction of that species. But that's that in itself, it is an impact, but not so much quantitative as we would have liked to demonstrate. But what we notice is, following up that introduction of that fish, we find that species of fish, which was very important in Europe, because it's one of the rare of that group in Europe displaying this kind of ecology, disappeared completely. And interestingly enough, it is a species which is very similar to Thomas Gudgeon's. It is very small, it's the same family, very small growing. Again, it has a very early maturity, it matures very early, it guards the nest, it guards the eggs, and it has very short longevity, so you could say it's a weed. But in fact, in Europe, and in most part of Europe, it's in danger or on the red list of species. In places it has even extinct completely. When it was introduced in England, it has been considered as invasive, although this is debatable. So we looked at this species, and when I was at CH, I did a little experiment in a pond, a large pond, where I put that European fish, and I put 1,200 fish in the pond. And a very simple experiment. And then just let them get on with it, and then after reproductive seasons, I just measure how many fish were there. And then after seasons, I saw that the number of fish have increased, like you would expect if the population is healthy, good recruitment, then the population growing. Then I decided to do a different experiment. So I removed all the fish from the pond and started again the experiment, but instead of having just one species, we kept the density constant, but we divided in between the two species, the Asian species and the European species. And the same, very simple experiment, let them get on with it and measure how many fish were produced after one season. And so instead of having an increase, we had a decrease of population. And then when we follow it up after 3 years, our population of European fish have completely gone extinct. So we were on something. We had demonstrated there was an impact of the introductions on that European fish. We didn't really understand what it was, and we had to go a bit further in our understanding. So I designed some experimental setup in the tanks, so inside the lab, to try to isolate what are the mechanisms of what we were observing. We set up a control where we had only the European fish here. And then we had a treatment where we had our European fish, but at the top we put our Asian invader, and they were just linked by water. The water was recirculating. They couldn't get in contact, so there was no direct competition. And we did a third treatment where we had the same setup as a treatment, but in between we sterilized the water. So the water was sterilized between the two. And then what we did, again, is very simple. We measure the number of eggs produced after a season and the mortality. After a season in the control, you got about 1,500 eggs, mortality 16%. And then you look at the treatments, and then you realize there's no eggs, not a single spawning. Complete stop of inhibition of spawning, but a high mortality as well. And then we did the treatments where we sterilize the water in between, and then the number of eggs go back to the control, and then we find that the mortality was very low. So here we understand, and we're starting to isolate that for some reason, something was released by that fish into the waters and affected this one. And then when we sterilized the waters, we got rid of whatever was released. A little bit more works on it show that, in fact, the fish, when they were dying, they were getting emaciated, they were losing weight. And I was fortunate enough to have a colleague, a French-Canadian girl, who was working at CIFAS at the time, who saw some similar things. We did some histological slides that she found in salmons in North America. And it was a disease which created mass mortality of salmon in North America and has been responsible for 90% loss of stocks in southern aquaculture. And so we did some DNA testing on that disease, and we found that it was the same disease. Although it was not exactly the same strain, it was the same disease, and it was called the rosette agent. And we managed to isolate that disease from our population and start culturing it. We culture it on salmon cell lines, so on salmon type of cells, and on cyprinids, which is a different type of fish cell line. So we managed to grow and culture our disease, which helped us after understanding the implications a little bit better. What it is, it's a little eukaryotic parasite. So it's a little parasite which live within the cell of the host, which is the division in terms of evolutionary tree between fungus and animals. So it's really at early stage of evolutions. And it does live within the cell. It doesn't have any organ specific. It can be in the liver. You can find it in the intestine, even in the gonads. And it divides by fission. And then when you put it in fresh water, not in salt water, but in fresh water only, then you will have a division and what we call zoosporulations, the creation of spores. The spores explode, and then you got the flagellate spores, zoospores, coming out of it. So we basically understood the structure of that life cycle, but we're not quite sure about how resilient this stage was to temperature. And it's very important. As you see, the host we have, the host has no limit in temperatures. You find it in northern Poland, you find it in Algeria. But that disease, to be able to have a good understanding in terms of epidemiology, we wanted to know if that phase of the life cycle was sensitive to temperatures, which would have limited the potential expansion. So we did some experiments trying to grow that zoosporulation phase at different temperatures. We tried 4 degrees, which are kind of trout-type habitats, and to tropical 30 degrees. And for the experiments, what we looked at is the onset of the events of zoosporulations. And we found that there was no inhibitions regarding to temperatures. If you put it in cold temperatures, then after 6 days, you have an onset of zoosporulation. If you put it at 30 days, you also have an onset of temperatures, but of those sporulations, but after only 1 day. So in fact, what you do, you accelerate the cycles by putting it in warmer, but it still survives in all temperatures, and it still produces high numbers of zoospores. So what we demonstrate is that in the most fragile part of the life cycle, it's still not so much temperatures dependent. We tested it on our native species. We tested it on salmon. Here we have the control of salmon, so the ones that are not infected with the disease, and we tried to infect it with the different cell line that we grow the disease on, the ones on the carp and the one on the salmonids. And in both cases, we had a significant increase of mortality as a result of the disease. And then you could see here the disease very clearly when you look at the fish. I'm not sure you could see it very well, but you could see the white kind of cover here on the liver, and here in the tissues, it's kind of gram stained, and you could see the presence of the disease. So very, very, very significant in salmons, very, very strong application. We tested it on carps. We had also an increase in mortality. We tested it on roach. We also had mortality. But that was done in the lab. So although we have understood the spread of the host, the importation of the disease, we're starting to understand what is the impact of the disease on our species. We wanted to test that in a much more natural system, and we tried in ponds and tried to recreate the same things in the ponds. What we cohabited is our fish, our Chinese fish, with our native species in different ponds, and just as against, let it grow and see what happened. And after 3 months, there's no problems. We had the ruds, they survived fine. Carps died, and they were getting emaciated. Roach were all dead, all disappeared, and the bream were all dead. So we, again, showed the same phenomenon that we find in the lab. We went all the way back to the field and find exactly the same phenomenon. So this is a good example of an introductions which have big implications for the maintenance of biodiversity. The European fisher we were looking at is now on the red list of species on the IUCN. So it leads to an important coverage, sometimes a little bit too far. But with question in, this is a sun, so you can't expect anything different. But you have a question in parliaments also. And the reason why it was so important is that it has implications for the aquaculture industry, for the salmonid aquaculture, which is very important here in Scotland as well. And it had also implications for the recreational fishing, which is the main sport in the UK. So it represented really the completions of what you would say the worst case scenario in terms of disease introductions associated with an invasive species. But that was not enough. You know, politicians, they like to know pounds. This is what convinced them really. So when we started with a university in London to calculate how much it will cost them to eradicate these species, if they're waiting too long, then they started to react. We started to calculate if they wait for 10 years, how much that will cost them to how much it will cost to eradicate these populations. So they decided. At the time, I contacted the people at DEFRA and the Environment Agency, saying, well, there are populations near the Lake Windermere, and Lake Windermere is a beautiful lake, a high conservation status and value. And if you introduce if the species get there, this is it. You will not get rid of it, and you don't know the implication might be great. That took a bit of time, but it trickled down, in fact, to the first eradication program in the UK. For the first time, the Environment Agency have decided to put in place an eradication program using chemicals to poison the lakes to be able to get rid of that. So it was a big milestone, and in terms of public communication, was a big step forwards for the Environment Agency to take. So they started to work on the lakes near the Lakes Windermere. It's called Rother-East-Tarn, and it's a little lake, little fishing lakes, a few hectares. And as you could see, Thomas Gozlan was extremely abundant in the lakes before. People come and fish all the time, so they feed the fish, and this is ideal condition for them to grow. And so what the Environment Agency used is a chemical called rotenone. Rotenone is a chemical extracted from a plant. It basically lower the oxygens in the waters and kill the fish by asphyxiation. So they remove all the larger fish within the lakes, and they quarantine them somewhere so they could reuse them later on. They lower down the level of the lakes, and they're starting to pump up the chemicals within the lake. And these are how they did it. So the idea is to have a good mixing of the chemicals, so you have all the water columns full of these kind of chemicals. And they finish up all the edges by hands walking along. And it was very effective, as you can see, and most of the fish died where Thomas Gozlan, so it was very successful in that respect. And after that, they put back the water's level. They reintroduced the fish there, and you could see carps reproducing very well. And so far, we haven't found the fish again in the lake. I mean, that was a very successful first attempt. And since then, they have developed a big programs of eradication all around the UK and been very successful at doing so. And as you remember in the graph, we're just in the colonization process. So they managed now to limit at least the expansions of Thomas Gozlan in the UK. So just to synthesize what I've been presenting here is that although accidental, the introduction of a little fish like that could have a particularly big issues. It could really have a large impact on our systems. It could also, if you look at, because people say, oh, it's accidental, we don't know where they come from, we can follow them because they're accidental. But in fact, they mirror some control introductions, like the grass carp in Europe. So if you understand the path of the controlled ones, you can follow the others. And the same way, you can, and this is also something that we learn on the way, is that it's not because it has been introduced in low numbers that it has lost its genetic diversity. It's in fact, it has still a very high genetic diversity, sometimes even higher than in native range. So when it has been introduced in places, after 4 years, in general, people detected it. But there was a very big variation in between countries. So what we need to put in place is early warning systems that, as soon as possible, we can detect the introduction of fish. And we know them because we know the pathways, we know how they get there. Disease spread is still remain the main risk associated with the introduction of species. And as we've seen with our diseases which have been, for example, for eels, which have been also very, very important in terms of decline of native stocks. And so although the species is very little, it could have very, very serious impact in the food web structures. It is not because it's a big predator that is going to necessarily have an implication of the food web. Although little, because of the sheer numbers of them, then it could have a very strong competition with our native species. And in general, this is more for my colleagues in my field, we need less anecdotal understanding of impacts. Although it's easier to claim impact when you have a big fish with a large mouth and big teeth, we need something a little bit more deeper understanding on what is the real ecological impact following the introduction of a species. We need this evidence because we need to build risk assessment. Politicians want that. Managers need that. They need to be in place so we can limit the damage for further introductions. We need to understand the biogeography of the species. What is, as I remember when I say, if we introduce them from Taiwan, what's going to happen? Maybe they can go even further. They can even colonize a part of Africa. So there's big implication of understanding what you're dealing with, not just the species, but the genetic. And last but not least, very crucial that we engage with monitoring how it will cost if we do not do something about it, because that's what makes our policymakers react and take actions. And I say that because I know that more species will come. These will come from China, but they may also come from different parts of the world. And these are species which exactly follow the same pattern and the same direction as the ones we had studied. So we have to keep an eye well open for these to come. Right. Now enough about fish. Yeah, I'm sure you're bored to death. So I'm going to open up a bit of talk and talk about world preservation, because I think this is where, as you can see, I'm passionate about my fish, and if I want the rivers to be there and in good state, and for that I need to make sure that I understand what are the pressures on water. Remember this map. Already, this is the incident water security issues, and this is the biodiversity threats. And you could see, I mean, it's not very nice because there's more red than anything else, and the red are the highest level of the scales in biodiversity threats. So these threats and these kind of pictures is today. We are 6.8 billion people on this planet. These are the threats today. Imagine by 2050, when we'll be 9.5 billion, what that's going to be. We cannot stay and watch that happening. We need to start to be proactive and think about it, how we can address these issues, because that's not going to get better if we stay like that. Mainly because the lifestyle of the world is very different. This is a lifestyle that maybe it's a little bit romantic, the pictures I selected, but they are from our trip in China. But really, China is a fantastic country. I really love the place. The rivers there are massive, and the rivers are like the vein within the society. They bring the food which sustain the entire economy of the place. Everybody's collects freshwater fish, and the fish is a big part of the food and the meal there. And so all the economy start up from there. You go early morning, you'll see everywhere fish markets like that. So it's a real part of understanding the society there is understanding. And it's a very rural type of living still in places. In contrast, we got our way of living, yeah? We have a very processed type food, large houses, big cars, and that's the kind of lifestyles we want. Some people say, well, the American dream is over. Let's slow it down, let's be sustainable, not have a big cars, but in fact, does it work really? And I'm saying that because when you go back to China, these pictures there were taken on the border with North Korea. Here you got North Korea, and here we are on the Chinese side. And here already you could see they want the Kentucky Fried Chicken, so why not? It's good. Tesco, they're available, why not? This is a very easy way of shopping. So this is not going the other way. It's going the same way as our society have developed. And that's the way it's going to keep going. So some people have calculated that. It's a fantastic way of calculation. I haven't looked at it in the depth of calculation, but I'm sure you have all heard that. So if all the developing countries have the same lifestyle as we have, we will need 2.5 Earths. Yeah, I like that way of looking at it. But I think this is a very dangerous way of looking at things because it doesn't focus your mind. Because in fact, what we do is only have one Earth. So what does that tell us? What does that tell us? It's not expendable. We are expanding our populations, but the resources are not expendable. So that tells us basically more of that. This is where it's going to. So we're not talking about pretty flowers and butterflies and pandas. Here we're talking about conservation in terms of preserving peace throughout the world. So what are the consequences of that? Well, basically our technology and medical research have changed this ecological balance. We are not in an ecological balance with the world. We are completely unsustainable. Our population is much too high for the resources. So we are there because of our technology. What is the way out? Well, the way out is probably using also our technology to come out of these equations, to maintain, to win that battle, and to rely on technology to allow us to win that ecological unbalance that we have at the moment. This has got nothing to do with it, you will say. Yeah, it's probably got nothing to do with it, but it's very interesting because I think it's an analogy that I want to take a bit forwards to understand the conservation ecology. Have you ever thought about why we have only 2 sexes, male and female? Not why many sexes, why not several sexes, 7? Why not a unisex where all the animals could reproduce with each other? Well, there is a very important theory, it's called disruptive selections. Just very quickly talk about that. Here we got the frequency of the parental type. So, for example, we got the gametes, yeah? The gametes produced by species. Very large gametes here, which very energy demanding, a lot of energy to put in, but they can sustain the embryo, so they can develop life within it. On the other hand, you have very tiny gametes, like male gametes, for example. You can produce them, very cheap to produce, not men's, but the gametes, yeah? A lot of them, but you can't grow life. Life is not enough energy there to grow the life within it. In the middle, you got something which is neither good for growing life because it's too small and still very costly, and you can't produce very much because, as I say, energetically, it's still costly. So selection applying to that, you end up by having only 2 models which would function. The ones which are very expensive to produce but can grow life, and the ones which are very cheap to produce but cannot grow life. Well, what has got to do with conservation? You will say, well, I'm not sure. But really, when you look at it in a conservation perspective, you could say that's the same things. We have 2 types of projects we could develop. The small-scale project, conservation initiatives, where we have focused our mind for so many years now, which have a potentially limited growth, which is sustainable farming and this kind of local scale developments to be in balance with our ecosystem. They're cost limited, but they are very local. I think the future is here, female type projects, where we got technology-driven conservation initiatives, where we put a lot of money in it to produce them, but then they have a very strong potential for economic growth. And you know that's what drives our societies, economic growth. So this is development, conservation with the potential of economic growth. Cost is high, but the scale is global. So what kind of technology-driven conservation there is? Well, maybe you would have expected me here, or maybe you wouldn't, as an ecologist. But GMOs is one way forwards. I know we have a lot of issues and a lot of debate, quite often misinformation about GMOs, but this is one way to go forwards. And I'm going to show you how few examples of GMOs could help my fish in my rivers to be conserved. Here, this is a salmon. Everybody's have maybe tried salmons before, and if you did, and if you're unlucky like me, it's probably a farm salmon that you got. And the farm salmons are heavily fed by wild caught fish. They're catching a lot of small prey at sea, processing them in pellets and feeding these fish. So there is a very big waste of overfishing of these species to be able to produce few kilograms of salmons. Now, Aquabunty Technologies, it's a Canadian company, have developed a genetically modified salmon. You see, same age salmons, but it grows 3 times bigger. So by growing a fish much faster on the same resources, you're basically reducing the input of fish that you're taking from the sea. So it's a direct effect on overfishing just by producing that kind of salmons. And it will soon become available in the market, but this have direct consequences on preserving the overfishing issues. Another aspect is the kind of crops we're growing. Yeah? In countries where, and you've seen the map I showed you before, where the red was, in a lot of countries water is scarce and there are limitations on water. And they're growing crops which are really demanding, highly demanding on waters. And there are limitations about the waters available. And so now the Americans have developed this kind of drought tolerance maize, and they're going to start the trial in September. And I mean, they're actually starting this summer. So what we have here is also, again, direct impact on our rivers and our fish, because as we grow these crops, we don't need as much water. We are reducing our pressure on our system. And by doing that, we allow still the same productions, but with less damage on our environments. And I say that, I just picked up that newspaper last week. It's a French newspaper, it's called Le Monde. And it says that in the last 30 years, the climate change stopped the productions of cereals around the world. It said it has reduced by 5.5% the wheat productions, and it has reduced the maize productions by 3.8%. So you see, we're already in crisis of, we want producing more, not less. And we're already in this kind of struggling to produce what we used to produce before, just because of the changing of climate. So this is a way of dealing with this issue. You might think, again, I go very far from the fish because I'm going to talk about pig now. But pig is directly related to fish. It's related, if you go across the channel and you go to Brittany, it's very interesting to see. It's a very big pig farm there, very intensive productions, lots of pigs. But pigs are not very good at one thing, is digesting phosphate. They're not good at that. So they're eating a lot, but they produce a lot of phosphates. And phosphates are either used to fertilize the field, or they get drained into the rivers, and they go. And by going into the river, big algal bloom, big production of brown algae, and basically a change in the structures with our rivers, and decline of salmon. Salmon population in Britain are just struggling, really. So what they have developed is this beautiful name called Enviro Pig. Yeah? This is what they are. It is pigs which are better, and they have been modified to digest better the phosphates. So the production, by doing that, by producing that kind of fish, you cut by half the phosphate that you would have in your rivers. This is very important. It's for us as conservation ecologists to engage with this kind of thinking. Not let them do Terminator genes in seeds banks. That's not what we want them to do. But we want them to think about developing the next lots of technology which have a direct benefit for our issues of conservation. I think that's a very strong importance for us to engage with it. And finally, I'll wrap it up by you, because I know you were expecting where you were coming in the equation. But you are the essential part of the equations as citizen and as an actor of these conservations. As I say, without water, there are no rivers, and without rivers, there are no fish. And what we want to understand is what can we do about it as an individual. Say, sign of scarcity. Well, this is Spain. This is Doñana National Park in South Spain. They're producing you strawberry that you like to eat when you watch Wimbledon. Yeah? These are the strains of water. Massive production of strawberry there. They don't have that much water there. Yeah? A big sign of scarcity there. This is heartbreaking. This is one of the 4th largest lakes in the world. This is the Aral Sea. It's between Kazakhstan and Uzbekistan. And they have diverted the waters to produce cotton. Uzbekistan is one of the largest producers of cotton in the world. I'm sure that would surprise you. Now look at the state of it. No water. The boats are the screen. It's absolutely dramatic picture. Again, And this is, I need to read that because it's so about 1,400 million cubic meters of water per year to produce soybeans in Brazil to export just for the UK. I'm not talking about the overall exportation, just for the UK. This is about 0.5 million Olympic swimming pools. This is to feed the cows to produce our hamburger. So this is the kind of scales we're dealing with, and that's what the issues we're dealing with. But your water, you could put in your new equipment, both at BNQ to reduce showers, sprinkling, or the push in the loo. But in fact, this is only 3% of your water consumption. The 97% are when you go shopping. This is where your water footprint is invisible. It is around 3.5 liters a day for the agricultural products you have. It is about 1,000 liters a day for the industrial product. This is your main consumptions of water. We don't know that here. We are in a country we are very rich in water. We don't really know that. But in the UK, 62% of our water footprint is outside our borders. We have a responsibility. We don't have any issues with water here, but we have a direct impact globally on the global scarcity of water. Just give you an example very quickly. 40 liters to produce one slice of bread. 180 liters to produce a kilo of tomato. 1,500 liters for a kilo of sugar. 3 million liters for 200 kilo of beef. 15,000 liters for a kilo of beef. 2,500 liters for hamburger. Don't tell me you don't feel guilty now. So what can we do? Well, we can ask our politicians to have a footprint assessment in the national water policy making. We need a coherence between the different policies and between waters, government policy, and environmental, agricultural, energy, trade, and the foreign office. We need to integrate that better. But we need also better organizational distributions of water footprints. No need to produce this kind of heavy crops in places where water is scarce. Let's be sensible about how we do that. And we need transparency, so as a consumer, we can buy in all knowledge. And if you feel like it, you can also adjust your diet. It's important. We have a role to do that, and we have to think. We have to be educated customers. We cannot just go ahead with that. So remember that conservation is less about panda. That's for my kids, just to have a good laugh. But it is more about revolution, national security, because that's where we're heading if we're not doing something seriously. So I just wanted to thank you. Yeah. And I wanted to thank also a lot of people who have helped me putting all this data for all these years together. Thanks very much.